RT-PCR is a laboratory technique that stands for Reverse Transcription Polymerase Chain reaction. It is a highly sensitive and reliable method for detecting, amplifying, and quantifying specific messenger RNA, mRNA sequences from a sample. This makes it an essential tool for detecting RNA viruses, studying gene expression, and diagnosing diseases. Step 1. Reverse transcription. The process begins by converting the unstable single-stranded RNA template into a more stable double-stranded complementary DNA, cDNA molecule. An enzyme called reverse transcriptase is added to the RNA sample. The reverse transcriptase uses the RNA as a template to synthesize a single strand of cDNA. The original RNA strand is then degraded. A second DNA strand is synthesized to produce a stable, double-stranded cDNA. Step 2. Polymerase chain reaction, PCR. The newly created cDNA is then amplified exponentially using the standard PCR process. This process involves a series of temperature cycles to create millions of copies of the specific DNA sequence. Denaturation. The double-stranded cDNA is heated to separate it into two single strands. Annealing. The temperature is lowered to allow short DNA fragments called primers to bind to their specific starting points on each single strand. Elongation. The temperature is raised again, and an enzyme called DNA polymerase extends the primers, building a new complementary strand of DNA. This cycle repeats 25-50 times, with the amount of targeted DNA doubling with each cycle. Real-time RT-PCR, RT-qPCR. While conventional RT-PCR provides results at the end of the process, a variation known as real-time RT-PCR, also called RT-qPCR, allows for the monitoring and quantification of the amplification process as it happens. Fluorescent markers. This method adds fluorescent dyes or probes that emit a signal when they bind to the new DNA copies. Real-time monitoring. Specialized equipment measures the increasing fluorescence after each cycle, which correlates with the amount of DNA being amplified. Quantitative results. This allows scientists to not only confirm the presence of a genetic material but also measure how much of it was originally in the sample. This is how a cycle threshold CT value is determined in COVID-19 tests. Common applications. RT-PCR has numerous applications in medicine, research, and diagnostics. Viral detection. It is the gold standard for diagnosing viral infections including COVID-19, SARS-CoV-2, by detecting the virus's RNA. Gene expression analysis. It is used to quantify the amount of specific mRNA in a sample, helping researchers study how much a gene is being expressed. Cancer research. It can detect changes in a gene or chromosome that may indicate a disease like cancer. Pathogen identification. It helps identify various bacterial, fungal, and parasitic infections.